Good evening, everyone. So this is Kanika Gupta, your TA for this session, and I'm a PhD scholar at IIT Delhi. So I'll be taking this session for you all. Those who have attended the last lecture must be knowing me, but it's okay. Let's start with the lecture. Um, in the last lecture, we discussed about the week one lectures what sir had provided you all regarding the structure of these prokaryotic and eukaryotic cells. What are the function of the different organs present in the prokaryotic and eukaryotic cells? So in the last lecture we studied about that. Now when we have a complete cell, when the cell is present, we have all the structures, we know the function of each and every structure. Now what's next? The next part is the metabolism, the different type of reactions that is going on inside a cell. Either it is the formation of some biomolecules or the degradation of some biomolecules for our requirements for energy production. So there are many kind of processes going on at the same time in a cell, and all of these processes together called as the metabolism. Okay, metabolism is further divided into two parts, the catabolism and the anabolism. So I hope you all remember, or you all understood the function of different cellular structures and what are the structures present in a cell. Either it is a prokaryotic cell or a eukaryotic cell. Further moving on with the catabolism and anabolism parts. So as I have briefed you already, that metabolism is divided into two parts, the catabolism and anabolism. Catabolism is basically the degradation, or we can say uh, simpler forms, the formation of simpler forms of any um complex biomolecules, so that it can be further used to generate energy from that molecule. So this is that part of the reaction, or that kind of reactions are called as the catabolism. The anabolism is basically the formation of complex molecules that is required by a cell for different kind of purposes is called as the anabolism, for growth, for for formation of new cell and anything that needs these complex molecules is all of these processes come under what? Anabolism. So this is what the basic difference between a catabolism and an anabolism. Next part is the when we go for the catabolism, there are different kind of molecules which have been categorized as either carbohydrates, lipids, or proteins. So these must be degraded down, or we can say it should be simplified in simpler molecules which can be further used, as I have already told you, to generate energy. So first of all, we'll see what is there in the metabolism of a carbohydrate. So carbohydrate we are getting from different food items, which when we eat, it enters through our stomach to stop small intestine and then to large intestine. When the food is in the stomach and small intestine, it undergoes different kind of processes. So the complex carbohydrate molecule is further simplified into the glucose molecules. These glucose molecules are then absorbed from the small intestine into the in the blood, and from there in the blood it is further decided whether it should be stored inside the liver in the form of a glycogen, or it should be further utilized to generate the energy depending upon the requirements of the cell, requirements of the body, or depending upon the various kind of signals present inside the blood. There are various receptors involved in this, which tells us whether this glucose should be stored in the form of glycogen inside the liver, or it should be further degraded down for the energy production. Many kind of signals are involved in this. That is a very detailed something that you guys must understand when reading the physiology part, the human physiology or any other animal's physiology. Here we'll discuss only how this carbohydrate is metabolized and further simplified to the level of the glucose, and then used further for the generation of energy. Sorry for this. Yes. So, first of all, the breakdown when this complex carbohydrate is further simplified to glucose, and then this glucose is further degraded down for the formation of energy. So the process of generation of energy from glucose, or simplifying the glucose from glucose molecule to pyruvic acid, the process is called as the glycolysis. So if we'll go by the definition, what is glycolysis? Glycolysis is central to carbohydrate metabolism, and it is the universal pathway found in prokaryotes or eukaryotic cells. It is a breakdown of six membered glucose into two three membered carbon sugar. क्या होता है इसमें? Glucose is a six carbon molecule. It should be broken down into simpler three carbon molecule, which then further enters the Krebs cycle, and another process, or we can say another reaction going on to fully utilize this glucose to generate maximum of the energy that can be generated from a single glucose molecule to its full potential. Okay, so it is what it is the breakdown of six membered glucose into two three membered carbon sugar to feed Krebs cycle in the presence of oxygen. Now it goes both ways. Either it can be in the presence of oxygen, or it can be in the absence of oxygen. In that case, the production of the amount of energy produced from both of these processes will be different. Okay, so when in the presence of oxygen, or to send for an anaerobic oxidation, or we can say the absence of oxygen. Okay, in the presence of oxygen is called as aerobic, in the absence of oxygen is called as the anaerobic. Hence, it plays a crucial role for adaptation of a living organism under different types of stress conditions. The glycolysis is a 10 step. You must remember this, that this is a if any one of you is preparing for some competitive exam or something. So let me tell you this thing, that you must remember each and every step of glycolysis by heart. At what step the energy is being used, at what step it is being produced. Either it is being formed in the form of NADPH, or it is being in the form of ATP. What step is called as what, and what enzyme is involved in which step. So you should know this glycolysis by heart. Okay, so glycolysis is a 10 step chemical reaction to enable glucose for its optimal oxidation and to produce maximum amount of energy that can be generated from a single glucose molecule. Okay, so the steps involved in glycolysis are, just a moment. So the steps involved in glycolysis are: step 1, as I told you, it is a 10 step reaction. So step 1 is phosphorylation of glucose. Basically, it is the activation of glucose molecule so that it can be further utilized by different kind of enzymes present involved in glycolysis. Okay, so glucose produced after glycogen breakdown. Glycogen, as I already told you, is the storage form of glucose that which is stored in the liver. So glucose produced after glycogen breakdown is phosphorylated by the enzyme glucokinase, या फिर हम इसको क्या कह सकते हैं, hexokinase. Either it can be called as glucokinase or hexokinase. If we are talking about it in the liver, then it is called as the glucokinase, and in all other tissues it is called as the hexokinase. Okay, especially in muscles. So in the phosphorylation reaction, what happens is, now we need to add a phosphate group to this glucose. Where from this will come? It will come from an ATP molecule. Only an ATP can provide this phosphate group, so that this glucose can be phosphorylated. And also this step requires energy, so an ATP molecule will be utilized here. It will form the ADP, and the phosphate that will be released by the the pyrophosphate, the utilization of this pyrophosphate, that is the triphosphate part of the ATP, then this phosphate released will be used to phosphorylate this glucose. So one ATP molecule is being used here. Till now we haven't generated a single molecule of ATP. We are using first molecule of ATP at this first step of glycolysis. Okay, so the group of the phosphate group of ATP is transferred to glucose to form glucose 6 phosphate, and the phosphorylation reaction of glucose to produce glucose 6 phosphate marks the molecule for glycolysis. It is the starting of glycolysis. Okay, and one molecule of ATP is utilized in this step. Okay, just I'll just start the pointer. Where is the cursor here? Let it be. Yeah. So, second step. What happens in the second step is the conversion of glucose 6 phosphate to fructose 6 phosphate. Okay, this is a reversible step. So this reaction is maintained in equilibrium, as you can see it from the diagram also. So in this step what happens is conversion of glucose 6 phosphate to fructose 6 phosphate. Till now it is a six membered carbon molecule which is proceeding further. We haven't came to the stage till now in which this glucose molecule, the six carbon glucose molecule, will be converted to the three carbon carbohydrate molecule. Okay, sugar molecule basically. So what happens in the step 1, what happened is the conversion of glucose to glucose 6 phosphate in the presence of enzyme hexokinase. In second step what happens is the glucose 6 phosphate is converted to fructose 6 phosphate, and that happens in the presence of phosphohexoisomerase. Yes. So it happens in the presence of what? Phosphohexoisomerase. Okay, so phosphorylated sugar produced in step 1 is converted into fructose 6 phosphate by the action of enzyme phosphohexose isomerase. But इस step पे we haven't used any ATP or any kind of energy molecule. Okay, so we have used only one ATP till now at step 1 only. Okay, next step is the phosphorylation of fructose 6 phosphate. Now, a phosphorylation is happening here. So, again, we'll be needing a phosphate group, and it will come from what? An ATP molecule. So, one more energy molecule will be used at this step. Okay, so this will be easier for you to remember. Whenever a phosphorylation is happening, ATP molecule is being used specifically in case of glycolysis. It is not an exclusive step, so you guys must not confuse this with anything else. Here we are just saying it for the sake of your learning, that whenever a phosphorylation or a phosphate group is being added to the six membered carbon molecule, an ATP molecule is being utilized, because the phosphate required for the phosphorylation of this molecule will be coming from ATP. And also these steps require energy, so this ATP will provide energy and phosphate group both. Okay, so at step 3 what happens is the phosphorylation of fructose 6 phosphate. In this step sugar is further phosphorylated at carbon 1 to produce fructose 1,6 bisphosphate. So at this step, this is step 3. At this step we have got two phosphate molecule, and we have utilized two ATP molecules. Okay, by the action of enzyme phosphofructokinase. Okay, so it is the enzyme phosphofructokinase. एक minute. चलिए. Now In the phosphorylation reaction, again, the gamma phosphate group of ATP is transferred to phosphorylated sugar to form fructose 1,6 bisphosphate, and one molecule of ATP is utilized in this step also. Okay, so to remember the enzymes, what you must remember is the molecule that will be forming. For an example, in case of glucose, when the glucose was being converted from glucose to glucose 6 phosphate, it is an hexose sugar, so the enzyme involved was hexokinase. Now, when this glucose 6 phosphate was converted to fructose 6 phosphate, so again it is a hexose sugar and it is a phosphate attached sugar. So, phosphohexose isomerase, because still it is a 6 membered carbon ring, but it is getting isomerized to form fructose 6 phosphate. So, phosphohexose isomerase. Now, at next step, what happens is first fructose 6 phosphate is being converted to fructose 1,6 bisphosphate. So, kinase, you must remember this, that the kinase is whichever enzyme has a kinase in it, it is involved in the phosphorylation. It will add a phosphate group to the succeeding molecule. Okay, so whenever a kinase is involved, as you can see in hexokinase, kinase was involved, and phosphofructokinase also kinase is involved, so it will indicate the phosphorylation. So phosphofructo, now this fructose 6 phosphate has a phosphate on that, so it is phosphofructokinase. Okay, now the molecule that we got is fructose 1,6 bisphosphate. Okay, so this was the step 3 and we have utilized two molecules of ATP till now. Next step is step 4, that is the cleavage of fructose 1,6 bisphosphate. Now, this 6 membered carbon molecule will break down to give us two 3 membered carbon molecules. Okay, so this is the breakdown step. Sorry, this step is catalyzed by the enzyme aldolase. Okay, so the enzyme involved is aldolase, or we can call it as fructose 1,6 bisaldolase. Okay, to generate glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. This glyceraldehyde 3 phosphate is an aldose and this dihydroxyacetone phosphate is a ketose, a ketonic sugar and an aldehyde sugar. Okay, next is step 5. Now, the as you can see, both of these glyceraldehyde 3 phosphate and dihydroxyacetone phosphate are both in equilibrium, that is they can interconvert between each other. Okay, so step 5 is what? Interconversion of triose phosphates. 3 carbon sugar formed in step 4 undergoes internal conversion as glyceraldehyde 3 phosphate can readily be able to enter into the next step. Okay, so it can interconvert into either glyceraldehyde 3 phosphate to dihydroxyacetone phosphate, and same goes for dihydroxyacetone phosphate, and the enzyme involved is phosphotriose isomerase. Okay, so the enzyme involved is phosphotriose isomerase, and the ketose generated in step 4 is reversibly converted into glyceraldehyde 3 phosphate by this enzyme. Okay, so the next step is step 6, in which what happens is this glyceraldehyde 3 phosphate, it forms 1,3 bisphosphoglycerate. Okay, so it will form 1,3 bisphosphoglycerate. So here also 1 NADH is in this step, one molecule of NADH is produced after oxidation of aldehyde group of glyceraldehyde 3 phosphate with the help of enzyme glyceraldehyde 3 phosphate dehydrogenase. Okay, so here what happens is this NADH will be, sorry, yes, this NADH will be produced here. Okay, at the next step what happens is step 7. In this step what happens, the phosphate group from 1,3 bisphosphoglycerate is removed by phosphoglycerate kinase with an acyl phosphate group transfer to ADP. तो क्या होगा, अब यहाँ पे एक ATP का मॉलिक्यूल बनेगा. ठीक है, यहाँ पे 1 ATP will be formed. And then after at step 8 what happens is the conversion of 3 phosphoglycerate to 2 phosphoglycerate, एक isomerization हो रही है यहाँ पे भी. So in a two step mechanism, phosphoglycerate mutase catalyzes a reversible shift of phosphoryl group to form 2 phosphoglycerate. This is the conversion. ठीक है, तो एंजाइम इन्वॉल्व इज फॉस्फोग्लिसरेट म्यूटेज. ठीक है, नेक्स्ट व्हाट हैपेंस इज the dehydration of 2-phosphoglycerate to phosphoenol pyruvate. ठीक है, the enzyme involved is enolase. ठीक है, enolic reaction हो रही है यहाँ पे, so enzyme involved is enolase. So the enzyme enolase catalyzes the dehydration reaction to produce phosphoenol pyruvate, a compound with high phosphoryl group transfer potential. This phosphoenol pyruvate has a more potential to transfer its phosphate group than 2-phosphoglycerate. ठीक है, हाँ. In the last step, the step 10th, what happens is this phosphoenol pyruvate will again, because it has more potential to transfer its phosphate group, so it will generate an ATP and will get converted to pyruvate, and the enzyme again will be involved is a kinase. So it will be pyruvate kinase. Okay, so this is how this glycolysis goes. In the glycolysis is completed in 10 steps, and at the end we get an end product is pyruvate, which enters the Krebs cycle. Okay, so if we'll calculate the ATP production, or we'll form the balance sheet for this glycolysis, then what it says is at step 1 and 4 2 ATPs are used. Sorry, at step 1 and 3 2 ATPs are used, as we have seen. Then when two molecules of glyceraldehydes are generated, so it is the calculation for the carbon thing. The step 6, at what happens at step 6, NADH is formed, and each NADH when enters the electron transport chain, it gives 3 ATPs. Okay, so one NADH can give 3 ATPs, 2 NADH will give 6 ATPs, because if you remember, now we have 2 3 carbon molecules. We have shown the cycle for one only, but we have 2 3 carbon molecules. So this NADH, the production of NADH will be 2. So the total ATP formed will be 6. Okay. Now at step 7, one ATP is formed, as we have seen in the cycle, but again, as I told you, 2 3 carbon molecules are involved, so 2 ATP will be counted. At step 10 also we got 2 ATPs. At step 10, if you can see, when phosphoenol pyruvate is getting converted to pyruvate, we got 2 ATPs. So we have used 2 ATPs, we have generated 6 plus 2 plus 2 plus 2. कितना हो गया? 10. ठीक है, सॉरी, 6 plus 2 plus 2, it is 10. एंड दो मॉलिक्यूल्स ऑफ एटीपी हमने यूज कर लिया है, तो टोटल मॉलिक्यूल्स ऑफ एटीपी हम लोग को जो मिलता है दैट इज 8 एटीपीज. एट द एंड वी गेट इज 8 एटीपीज. Okay, now when we'll come to Krebs cycle, as I told you, this pyruvate molecule will enter the Krebs cycle. So what happens, first of all, what is Krebs cycle? So Krebs cycle is discovered by Professor Hans Krab, and as it has all sugar intermediates with 3 carbon, it is also known as tricarboxylic acid or citric acid cycle. ठीक है? In higher eukaryotes, Krebs cycle operates inside the mitochondrial stroma with the different enzymes, and in the presence of oxygen, pyruvate formed during glycolysis enters into the Krebs cycle for further oxidation to produce energy. Okay, so in the presence of oxygen, pyruvate formed during glycolysis will enter into the Krebs cycle, as I told you already. So for further oxidation and to generate more energy. So the pyruvate from glycolysis will enter to this Krebs cycle with the help of enzyme pyruvate dehydrogenase complex, and it will form what? Acetyl coenzyme A plus water. This acetyl coenzyme A along with oxaloacetate in the presence of citrate synthase will form the citrate molecule, again a 3 carbon molecule. This citrate will then convert it to in the presence of aconitase will form cis aconitate. The cis aconitate again in the presence of aconitase will form isocitrate. Okay, this isocitrate at every step, if you can see, Miss Divya Rai, can you please mute your mic? Better, thank you. So yes, this I as I was telling you guys. So when the citrate synthase was involved, so coenzyme the group that was involved in that was attached to acetyl coenzyme A, that coenzyme A along with the reduction group, that is the SH group, it got released from this molecule and the citrate is formed. Then when acanitase is acting on the citrate, dehydration reaction is going on and one water molecule is being released. When this cis aconitate is again converted to isocitrate, another dehydration reaction happened and one water molecule was released. Okay, now when this isocitrate was getting converted to oxalosuccinate, the NADH was formed. Okay, isocitrate dehydrogenase, it will lead to the formation of an NADH. Okay, the oxalosuccinate will get converted to what? Alpha ketoglutarate, releasing one CO2 molecule. Then this alpha ketoglutarate molecule will be converted to succinyl coenzyme A. The coenzyme A with this its reduced form, the SH group will enter the reaction and an NADH will be formed. This succinyl coenzyme A will then get converted to succinate. A GTP molecule is formed and this coenzyme A will be removed from here. Then this succinate will get converted to fumarate, forming a molecule FADH2 in the presence of succinate dehydrogenase. Now this fumarate will form the malate in the presence of fumarase, again leaving a water molecule, and this is again a dehydration reaction. Then this malate will again get converted to oxaloacetate, a reaction that is going on. So this is how this Krebs cycle works, and an NADH molecule will be released. Okay, sorry, will be utilized. So if we'll see the balance sheet again for Krebs cycle, so the production of acetyl coenzyme A, उसके बाद at step 3 generation of alpha ketoglutarate, again it is a number of ATP produced. So finally what we will get is the 30 molecules of ATP from this Krebs cycle, and 8 molecules that we got from the glycolysis. So in total what we got is we got 38 molecule of ATPs from a single molecule of glucose. So this much amount of energy is generated from single molecule of glucose if it degrades or if it breaks down uh, in the aerobic reaction. If we'll go for anaerobic reaction, the balance sheet will be different. Here we are discussing only about the aerobic reaction. So the molecules generated for ATP is 38. Okay. Okay. So how the regulation of Krebs cycle occurs? There are four rate limiting steps in the Krebs cycle, and the points where it can be regulated is conversion of pyruvate into the acetyl coenzyme A, जो बिल्कुल first step था, when the pyruvate from glycolysis is entering the Krebs cycle. So at that step, sorry, as it is a rate limiting step, so that can be the reaction can be controlled, regulated there. So conversion of pyruvate into acetyl coenzyme A is the first step which allows the entry of sugar moiety into the Krebs cycle. The pyruvate dehydrogenase complex is allosterically inhibited by high ratio of ATP. When we have ample amount of energy present inside the cell, then in that case this the high ratio of ATP to ADP, that means that when we take a ratio of the presence of ATP divided by the ratio of ADP, when this ratio is high, that means when the ATP amount of ATP is high, that inhibits the high amount of ATP allosterically inhibits the pyruvate dehydrogenase complex. Allosteric inhibition matlab it won't bind to the active site, but it will bind to somewhere else near the active site, which will change the shape of the active site, and that is how the complex won't be able to function. Okay, so that is how the allosteric inhibition works. So the high ratio of ATP to ADP, NADH to NAD positive, and acetyl coenzyme A to coenzyme A will actually inhibit this pyruvate dehydrogenase complex, and it's entering further into the Krebs cycle. And if it won't enter the Krebs cycle won't be able to proceed. Okay. So the first reaction of Krebs cycle catalyzed by the citrate synthase is inhibited by high levels of NADH, ATP and succinyl coenzyme A. So another step which is at which the Krebs cycle is regulated is the conversion of citrates at the functioning of citrate synthase, where the high levels of NADH, ATP and succinyl coenzyme A will inhibit the enzyme and will stop the cycle. Okay. Next point where the cycle is regulated is the isocitrate dehydrogenase, which is inhibited by high levels of ATP NADH, whereas calcium and ADP stimulate this step. If ADP is present in higher amount, that means the energy is low, and if the energy is low, that means the cycle needs to run to produce more energy. So in that case this isocitrate dehydrogenase will be stimulated. If the amount of ATP is high, that means the energy is high. In that case this isocitrate dehydrogenase will be inhibited. Okay. Next step at which the regulation of Krebs cycle happens is alpha ketoglutarate dehydrogenase, which is inhibited by succinyl coenzyme A and high levels of NADH, whereas calcium stimulates this step. Okay, so why calcium stimulates this step is, I'll just give you a very small brief. So calcium is actually involved in many kind of the activity related reactions, or we can say the actions happening inside the body. So when the calcium will be there, calcium will come into action, as I told you, when body is in some kind of action or it is some kind of movement. So those steps will require energy. So presence of calcium acts as a stimulus, because the presence of calcium stimulates these enzymes that the energy is required. Whatever energy is left will be utilized and more energy will be needed, so you start producing energy. In that case, the cycle will run as it should. So that is why calcium acts as a stimulus to stimulate these steps and for the proceeding of the Krebs cycle. Okay. Okay. So Krebs cycle is also called as the master regulator of metabolism. Why it is so? Because Krebs cycle is centrally connected to the metabolic intermediates of the carbohydrates, proteins, and lipid metabolism. Okay, it has several branching points where it can communicate with either protein or lipid metabolism. So, lipid metabolism is connected to Krebs cycle through common intermediate as citrate and acetyl coenzyme A. If you guys have listened the lecture carefully, I'll just give you a brief about that also. That whenever the beta oxidation happens, the end product that is formed is acetyl coenzyme A. Okay, so that is how the lipid metabolism is connected to Krebs cycle through common intermediate such as citrate and acetyl coenzyme A. Next is the protein metabolism. Protein metabolism shares intermediate at alpha ketoglutarate oxaloacetate. As a result, Krebs cycle can allosterically or through product inhibition regulates other metabolic pathways also. Okay. In addition, it can redistribute intermediates between metabolic pathways and hence help in conversion of sugar to proteins, lipids, or vice versa. So intermediates of Krebs cycle are involved in many other processes also, in many anabolic reactions also. So that is why the Krebs cycle is the master regulator of metabolism. ठीक है. अच्छा, evolution में क्या role है Krebs cycle? So Krebs cycle is directly associated with running of electron transport chain and hence depends upon availability of the oxygen. Development of Krebs cycle has evolved the organism to adopt into the high oxygen environment, because initially when according to evolution, when world started, it was highly anaerobic. ठीक है, anaerobic था. But the development of Krebs cycle is one of the factors that has evolved the organism to adopt into the high oxygen environment. Now, the lipid metabolism. Here, till now, we were discussing about the carbohydrate metabolism. Now, the term comes for lipid catabolism. So, what happens in case of lipid catabolism? Fat, whenever we ingest fat in our diets, it gets, when it ingested inside our body and it enters the stomach, the triacyl glycerol, sorry, through stomach it enters into the small intestine. In small intestine, we all know the secretion comes from different parts, which also includes secretion from liver, and that is the bile juice. So bile juice, what does bile juice have? It has bile salts released from this bile juice. They act from the gallbladder. They are actually very important to emulsify the fats, which results into the formation of micelles and fatty acids. ठीक है, so fatty acids will be formed. Tanvi, please mute your mic. So yes, result in the formation of micelles and fatty acids. Now this fatty acid, what happens with these fatty acids, या triacylglycerols, are degraded by the intestinal lipases. Lipase matlab enzyme that will break down the lipid molecules. ठीक है. So these triacylglycerols are degraded by the intestinal lipases. The intestinal mucosa takes up fatty acids and other breakdown products to convert them into triacyl glycerols. Till now it is fatty acids. Then when they are taken up in, they are absorbed inside the walls of this intestinal mucosa. Lacteals is the structure which is present in the intestinal mucosa, which absorbs these fatty acids. They take up and break down the product to convert them into triacyl glycerols. Okay. Now, what happens is these triacylglycerols are packed with cholesterol and apolipoproteins in chylomicrons. Now, these chylomicrons traverse through the capillaries via bloodstream to different tissues. These fatty acids then enters the myocytes and adipocytes where they undergo degradation, या फिर beta oxidation we can say. Just beta oxidation. Now, after beta oxidation, then these fats are simplified into simpler forms, which can further lead to the formation of the energy. Okay, so now let's see what happens. Why beta oxidation? What is beta oxidation? So beta oxidation is the sequential removal of two carbon fragments from the carboxyl ends of fatty acids. During the process, acetyl coenzyme A is formed as the bond between alpha and beta carbon atoms are broken. Okay, so during this process, what happens is acetyl coenzyme A is formed as the bond between alpha and beta carbon atoms are broken. It is named so because the beta carbon of fatty acid is oxidized and the process occurs in mitochondria. So beta oxidation, where does beta oxidation happens? It happens in mitochondria. So I'm not going into the details of the beta oxidation step by step because I hope you guys have read it from the lectures only. We'll directly go to the fatty acid activation and transportation to mitochondria. I'll not discuss the beta oxidation in detail, but how these fatty acid molecules they reach the mitochondria, we will uh, revert that only. Okay, so the enzymes of beta oxidation, as I already told you, are located in the mitochondrial matrix. So the fatty acids which with chain length greater than 14 carbon, they cannot pass the mitochondrial membrane. As you guys remember from the first lecture, that the mitochondria is a double membrane structure. The outer mitochondrial membrane, the inner mitochondrial membrane. The inner mitochondrial membrane forms the cristae also, and there is the space that is enclosed by the inner mitochondrial membrane is called as the matrix of the mitochondria. Okay, so the 40 the fatty acids with chain length greater than 14 carbon, they cannot enter mitochondrial membrane as such. Therefore they first undergo activation and then transportation, for which enzymatic reactions are involved and enzymes are involved. Okay, so once the fatty acid reaches the target cells, their activation takes place in the cytosol. Fatty acid activation is an ATP dependent acylation reaction. Okay, so the reaction involves an ATP. Just a moment, class. Sorry for the interruption. So yes, fatty acid activation is an ATP dependent process in which fatty acid is activated by coenzyme A and ATP to form fatty acyl coenzyme A with the help of an enzyme acyl coenzyme A synthetase. Okay, enzyme involved is acyl coenzyme synthetase, which is also known as acyl coenzyme ligase or acyl coenzyme thiokinase. Because why thiokinase? Because the bond through which this fatty acid is attached to this acyl coenzyme A, is sorry to this coenzyme A is formed through a sulfur, and thio term is used to indicate sulfur. Okay, so this acyl coenzyme A can coenzyme A synthetase can also be called as acyl coenzyme A thiokinase. ठीक है. So this enzyme catalyzes the formation of a thioester linkage between the carboxyl group of fatty acid and the thiol group of coenzyme A to yield the molecule of fatty acyl coenzyme A. Now, what happens at next step? Each fatty acyl coenzyme A molecule is converted to fatty acyl carnitine derivative. This coenzyme A molecule will be further converted to carnitine derivative in a reaction named transesterification in the presence of enzyme carnitine acyl transferase 1, which is present in the outer membrane of mitochondria. It is still going on in the outer membrane of mitochondria. We haven't till entered the mitochondria. Okay. So the driver is the derivative is translocated to mitochondrial matrix via acyl carnitine carnitine translocase. ठीक है, या तो हम इसको एसाइल कार्निटिन कह दें या फिर कार्निटिन ट्रांसलोकेस कह दें, which is present in the inner membrane of the mitochondria. So there is a transporter involved which will take this acyl carnitine derivative to the inside of the mitochondria, that is the matrix part. Okay. So fatty acyl coenzyme A is regenerated via carnitine acyl transferase 2 located on the matrix side of the inner mitochondrial membrane. Carnitase is transported back into the intermembrane space via acyl carnitine transporter, which is then ready to participate in the next reaction, or we can say for the transfer of next acyl carnitine derivative. ठीक है. Okay. Stage 2 is what? The oxidation, the beta oxidation, as I told you. We are not going into the detail. Just for your revision, one thing you must remember is that palmitoyl coenzyme A, a 16 carbon fatty acid, when it will be oxidized, beta oxidation will occur. It will give us 7 FADH2, 7 NADH plus 7 protons. ठीक है. So these are the things that we are gonna get from the beta oxidation of palmitoyl coenzyme, and also 8 acetyl coenzyme A will also be provided. ठीक है. Next step is the regulation of fatty acid biosynthesis and catabolism. So what happens in this biosynthesis and catabolism? So fatty acid का हम लोगों ने देखा कि कैसे उसका ब्रेक डाउन होता है, हाउ इट एंटर्स द माइटोकॉन्ड्रिया, एंड आफ्टर एंटरिंग माइटोकॉन्ड्रिया हाउ इट इज डिग्रेडेड इनटू इट्स सिंपलेस्ट फॉर्म, दैट इज द एसिटाइल कोएंजाइम ए. This acetyl coenzyme A will further enter the Krebs cycle and will help in the generation of the energy. The FADH2 and NADH form will enter the ETS, that is the electron transport chain, to generate the ATP molecules. Okay. So in liver, what happens is this fatty acyl coenzyme A has two major pathways. It can either transport it to mitochondria, as we have seen earlier, through carnitine shuttle, so that it can get beta oxidation oxidized there, and it can convert it to acetyl coenzyme A molecules, or uh, sorry, it can get converted to triacylglycerols and phospholipids via cyto, or it sorry, the second step which the second thing which can happen with this fatty acyl coenzyme A is it can get converted into triacylglycerols and phospholipids via cytosolic enzymes. We also need phospholipids also because it is the core member of our membrane, the cell membrane that we have. So obviously either it can get oxidized or it can form the phospholipid. Okay. So the carnitine shuttle, uh, it has the rate limiting step for fatty acid oxidation and therefore is an important point of regulation. Once fatty acid are transported to mitochondrial matrix, they are destined for beta oxidation. if they have entered the mitochondria, beta oxidation will happen for sure. But if they haven't, then they can form the triacylglycerols or phospholipids. Okay, so the malonyl coenzyme A, the first intermediate of fatty acid biosynthesis via acetyl coenzyme A, also regulates fatty acid oxidation, as we have seen in case of Krebs cycle. Also, if the amount of malonyl coenzyme A will be high, then it will inhibit the carnitine shuttle, and if the carnitine shuttle will be inhibited, no more fatty acids or will enter the mitochondria for beta oxidation, and the process will come to an halt, or we can say it will lessen in its performance. Okay. Okay, another thing at which the regulation can happen is when the NADH to NAD positive ratio is high, indicating enough energy for cell to perform vital activities. So it will inhibit beta hydroxyacyl coenzyme A dehydrogenase, enzyme involved in beta oxidation. Okay. So high concentration of acetyl coenzyme A inhibits the thiolase. During the times of vigorous muscle contraction, strenuous exercise, or fasting, the consumption of ATP is increased, which reduces the concentration of ATP and increases the concentration of AMP, that activates AMPK pathway. Okay, so AMPK phosphorylates various other target enzymes such as acetyl coenzyme A carboxylase, which catalyzes malonyl coenzyme A synthesis. This phosphorylation and thereby inhibition of acetyl coenzyme A carboxylase brings down the concentration of malonyl coenzyme A. And if the concentration of malonyl coenzyme A will decline, so it will not inhibit the carnitine shuttle, and again the beta oxidation will start, energy production will start, and fat will be degraded. Okay, so these are some of the steps at which the catabolism is regulated. This is just what we have read already. uh, so I hope you guys must have understood it from the lectures also. Now, when the fat we have discussed for carbohydrate metabolism, we have discussed for fatty acid metabolism. Now the time comes for the discussion of protein metabolism. Okay, so protein is formed from what? Amino acids. Okay, so amino acids are divided into two categories broadly: the essential amino acids and the non essential amino acids. Okay, so the essential amino acids are those amino acids which cannot be formed inside our body through any kind of substrate or any kind of the initial molecule. The essential amino acids can't be synthesized in our body, but the non essential amino acids can be synthesized in our body. So what happens for essential amino acids? We are dependent upon our dietary. Sorry. Has the sharing stopped? Are you guys not able to see my? Now are you able to see the slides? I don't know how it stopped. Thank you, Srojini. I'm so sorry if I pronounced your name wrong. Okay, I will start from the left. So yes, I was telling you about the essential and the non-essential amino acids. So essential amino acids, we are dependent upon the dietary things for this. Non-essential amino acids is something we can form inside our body. How we can form them inside our body? Let's see. So, the biosynthesis of amino acids. Principally, all amino acids are derived from either glycolysis or citric acid cycle or pentose phosphate pathway intermediates. Not directly from these processes, but intermediates of these processes. Okay. So, these derivatives provide the carbon skeleton for the amino acids, whereas the amino group or nitrogen for the same is provided either by the glutamine or glutamate. Whose mic is on? Can you please mute your mic? Whoever it is, kindly mute your mic. Bhavna, I guess it is yours. Okay. So, what happens is, um, yes. So, we'll get the carbon skeleton from either glycolysis from citric acid cycle or pentose phosphate pathway, but the nitrogen or amino group required to make them a full-fledged amino acid will be derived from what? Glutamine or glutamate. ठीक है. Not all amino acids are synthesized by the organism, as I already told you. Those which we can synthesize will be called as the non-essential amino acids. Those which can't will be called as the essential amino acids. ठीक है. एक minute. ठीक है. So the most important reactions that take part in almost all the biosynthetic pathways of different amino acids are reductive amination of alpha keto acids. Reaction leads to the formation of glutamate. Transamination reaction performed by aminotransferase requires an enzyme PLP, that is pyridoxal 5 phosphate. So these are some of the must reaction that will happen in the pathway of every amino acid synthesis. Okay, chalo ji. So the overview of the amino acid biosynthesis is, as I told you, the glycolysis, the molecule from glycolysis which is involved is 3-phosphoglycerate. Okay, this 3-phosphoglycerate is involved in the formation of serine. The ribose 5 phosphate, which is formed by glucose, is involved in the formation of histidine. The 3-phosphoglycerate will give rise to erythrose 4-phosphate, is involved in the formation, along with the phosphoenol pyruvate, is involved in the formation of tryptophan, phenylalanine, tyrosine, all of the aromatic amino acids. Now, this pyruvate is involved in the formation of alanine, valine, leucine, isoleucine. Serine will derive the formation of glycine and cysteine. The oxaloacetate, which was present in what? In the Krebs cycle. It is involved in the formation of aspartate, which will lead to the formation of asparagine, methionine, threonine, and lysine. The alpha ketoglutarate, again from the Krebs cycle, is involved in the formation of glutamate, and this glutamate will lead to the formation of glutamine, proline, and arginine. Okay, the erythrose 4 phosphate and ribose 5 phosphate are actually the members of the pentose pathway. Okay. Now, this was about the amino acid biosynthesis. It was just to make you guys revise what we have studied in the last week. Now we'll come to the cell cycle part. Okay. So cell cycle. As we have read about the different structures involved in a cell, so there must be a proper way to replicate these kind of organs or organelles of any cell, so that every daughter cell can get equal amount of each and everything. One thing. Another thing is, if every cell has a fixed life. Okay, so these cells must divide to form the daughter cells, which can take place of the old cells, so that the organ as a whole won't get much older. And another cell cycle is obviously important for the procession of any kind of cell, and as it is, as the division and all of these processes are occurring in a cyclic form, that is why this division of cell is called as the cell cycle also. It is a very precise process. There are very many checkpoints involved at every step in this cell cycle, so that there won't be any kind of disappearances or any kind of, we can say, problems in the formation of either the genetic material or organelles of a cell, so that every daughter cell won't will get equal amount of each and everything and won't be defected. Okay. So what is cell cycle? The eukaryotic cell undergoes precise cell cycle and division to produce two daughter cells. Cell cycle is a series of tightly regulated events, that is the checkpoints, in events leading to the division and duplication. It is a vital process used to single cell fertilized egg is developed into full organism. If you guys have known a bit about the developmental biology, then you must know that whenever a male gametocyte and a female gametocyte fuse, it forms a single cell organism, a single cell after fusion, that is a zygote. Now this single cell develops into a full-fledged organism. How does this happens? It happens through the division of the cells, and this division must be tightly regulated so there won't be any kind of disappearances. Okay? So the process of the formation of these two daughter cells, the tightly regulated process involved is called as what? Cell cycle. So, cell division is the crucial event underlying the regeneration and repair in any tissue, liver, and for example, liver and heart. In prokaryotic cells, parent cell is divided simply by division into two halves through the process of binary fission. In case of prokaryotes, it is only the binary fission, but in case of eukaryotic cell, it is a much more complex system which is called as the cell cycle. So in eukaryotic cell, cell cycle has three phases. The interphase, in which the synthesis of genomic DNA and cytoplasm occurs. The mitotic phase, where the division occurs. And the cytokinesis, where the actual division of cell occurs and two daughter cells can be seen. Okay. Cell cycle, first of all, the interphase. The interphase is the preparatory phase required to perform requisite steps. These are series of events in the nucleus as well as in the cytosol of the daughter cell to enable it to enter into the division phase, and this phase has several phases. ठीक है, if we'll go by series, it is the G1 phase, the S phase and the G2 phase. ठीक है, G1 phase is also known as the growth phase. In this phase, cell prepares itself for the division. It checks whether I have an ample amount of nutrition, whether I have an ample amount of cytoplasm, whether I have an ample amount of each and everything which is required for division except for the genome, because genome will be duplicated in the S phase. Okay, so another thing which are required during a cell division or cell cycle will be checked in this phase only, whether I have the ample amount of each and everything or not, and if not, it will be synthesized in this phase only. Okay, so duration of G1 phase depends on the cell type within the organism. Okay. Next is the S phase. In the S phase, mainly the division of the genome occurs. The genome is replicated, and from 1x, its amount rises to 2x. It will be duplicated. Amount will increase here for the genome. Okay. Uh, So the synthesis of DNA occurs very fast to avoid exposure of newly synthesized DNA to any kind of mutagens. So this must be something that you guys must remember. And the G2 phase. This is again a growth phase between DNA synthesis and mitosis. During this phase, cell increases its size because now it has to divide into two cells. So cell increases its size and synthesizes the other proteins or cellular factors which are left to synthesize, which is required for the cell cycle. Okay, for the further division, for the mitosis and cytokinesis. ठीक है, G naught phase. अब G naught phase क्या है? So after G1 phase, sometimes cell enters the quiescent phase, or we can say uh, senescent and non-proliferative multicellular, the quiescent phase. So what happens is, after G1 phase, the quiescent, senescent and non-proliferative multicellular eukaryotic cell enters into the G naught phase. Cells remains in this phase for longer period of time till the time they get any stimulus for division. they'll stay in the G naught phase only. For example, in case of neurons always, once they are divided, they will stay in the G naught phase only, because neurons do not divide. Okay, so it is also common in fully differentiated cells. The fast growing cells never enters into the G naught phase because they keeps on dividing. Hence, it is not a regular cell cycle phase, and under specific condition, cells enters into the G naught phase. Okay. So what happens in the mitosis or M phase? After G2 phase, cells enters into the mitosis or M phase to divide the DNA equally between the two daughter cells. Each mitosis has four distinct phases: the prophase, the metaphase, anaphase, and telophase. ठीक है? Before cytokinesis, the M phase is further divided into the four phases: prophase, metaphase, anaphase, and telophase. So in prophase, what happens is the condensation of DNA molecule or genome. The duplicated genome will be condensed here, and it will form the chromosomes. ठीक है? So the chromosomes will be formed in prophase. In metaphase, what happens is a metaphasic plate is formed. The condensed chromosomes are arranged on a metaphasic plate at the center of the cell, so that every chromosome is arranged on that metaphasic plate. If you can see it here, this is the picture of a metaphase. The chromosomes are arranged in the in center in a single line as a metaphasic plate, so that the chromosomes are at equidistance from both the centers, and also all the chromosomes are aligned here, so that each daughter cell can get a single copy of each of the chromosomes. ठीक है? So, the alignment of chromosomes along with the metaphase plate is a crucial event to decide the entry of cells into the anaphase, and the signal required for this control is created by the mitotic spindle checkpoint. ठीक है? Next is the anaphase. What happens in anaphase is, now the chromosomes that are arranged at the metaphasic plates in the center of the cell will be pulled in opposite directions, so that the single copy of each of chromosome can be provided to each of the forming daughter cells. Okay? So the protein attached to each chromatid are cleaved, and the sister chromatids are separated as daughter chromosomes. And the chromosomes lined on metaphase plates are pulled by microtubule and move towards their respective centrosomes through the two centers. Okay? Although the exact mechanism of generating force required for centrosome movement is unknown, but it is suggested that rapid assembly and breakdown of microtubules may provide the force for this movement. And at the end of this phase, chromosomes are being prepared for distribution between two daughter cells. Okay? Now the last phase is telophase. In this phase, what happens is the daughter chromosomes have moved to their respective places, to their respective poles. The opposite ends of the cell, a nuclear membrane forms around each of these sets of separated daughter chromosomes, and nucleolus reappears. In this event, several processes during prophase are reversed to give two daughter nuclei. Okay? And after telophase, what happens is cytokinesis. Now the cytoplasm will be divided. So at the end of telophase, mitosis is over, but cell division requires distribution of cellular contents equally between the two daughter cells, and in animal cells a cleavage furrow is formed. Furrow means two opposite side projections, inward projections will formed along the metaphasic plate, and divides individual nuclei as separate cells. During this process, it is ensured that besides nuclei, all other cellular organelles should be distributed equally between the daughter cells. In plant cells, cell plate is formed, and which divides the cellular content between the daughter cells. तो plant cells में cell plate बनती है. In case of animal cell, cleavage furrow बनती है. ठीक है? So this is just an overview of how the cell cycle runs. Role of cell cycle क्या है भाई? It is important for the development of single cell into the multicellular system. is possible due to the cell cycle and division. Cell replacement में लिए basically repair के time पे, regeneration के लिए when some part is damaged, so to regenerate that part of an organ, cell cycle is important. Asexual reproduction जैसे कि binary fission that happens in hydra or other prokaryotic cells. ठीक है. हाँ. So cell cycle is controlled at different checkpoints which are present in the cells, in the cell. These cell cycle checkpoints are used to ensure that the completion of different steps and repair of DNA damage occurs properly. The main checkpoints are present at G1 to S transition, G2 to M transition, and at M phase, in between the metaphase, from anaphase to telophase. Okay? Each checkpoint is controlled by mutual interactions between cyclin and cyclin dependent proteins, kinase, basically cyclins and CDKs. P53 gene products are known to control many events through these checkpoints. ठीक है? If the any of these events goes wrong, then it can lead to the formation of tumors. Cancer basically is the undifferentiated and uncontrolled division of the cell, which happens when there is some kind of a problem during the control of the cell cycle. So this leads to the formation of the tumors. ठीक है? Alright. Okay. So we have discussed this already. Now a cell is formed, but a cell has a age also, and when the time comes, the cell must undergo a programmed cell death so that it won't lead to any kind of other mutations or any kind of discrepancies which can further lead to major kind of problems for an organism or inside the cell. Okay, so the programmed cell death is also called as what? Apoptosis. Apoptosis is a fundamental process in multicellular organisms which play a crucial role in various biological processes such as development, tissue homeostasis, immune responses, and the elimination of damaged or potentially harmful cells. Okay? So, apoptosis is a vital process that is happening inside an organism. Okay, it is a tightly regulated physiological process that rapidly removes damaged, mutated, or virus infected cells within an organism. ठीक है, मेजर फीचर्स क्या है एपोप्टोसिस के? इट इज अ कंट्रोल्ड एंड ऑर्डरली प्रोसेस. इट in contrast to necrosis, we'll see the differences between apoptosis and necrosis. Specific signaling pathways and molecular events derive the process. Cellular components are dismantled, leading to cell shrinkage. DNA fragmentation occurs, resulting in characteristic ladder patterns. And apoptotic bodies, basically the small vesicles, are formed. So these are the hallmarks of apoptosis, that a cell is undergoing apoptosis. Okay? Steps involved is cell will receive a stimulus. So this stimulus will lead to the shrinkage of the cell, the breakdown of cytoskeleton. Dense cytoplasm will become more dense. The packing of organelles will occur. Condensation of chromosomes will, chromatin will occur, and the fragmentation of DNA will lead to the formation of a ladder-like pattern and the breaking of the nucleus also. Then the formation of Bleb's cell breaks and formation of apoptotic bodies, the small vesicle kind of structures. And then the phagocytosis will happen. The macrophages will phagocytose this dying cell. Differences between apoptosis and necrosis is intact cell membrane blebbing occurs, while in case of necrosis the cell membrane is disrupted. Basically what happens in necrosis, it the in case of apoptosis, cell will not try to harm any other cell in its vicinity, so it will die in a manner that it will form small vesicles kind of structure, so that all of its material is present in those vesicles, and those vesicles will be ingested by the macrophages, so that the harmful material won't burst out and affect the nearby cells. While in case of necrosis, the cell membrane is disrupted, leading to the loss of membrane integrity. Begins with swelling, condensation of chromatids, followed by the dissolution of sorry the condensation of chromatid and fragmentation of nucleus occurs, followed by the dissolution of cytoplasm. Complete cell lysis occurs, no vesicles are formed. While in case of apoptosis, vesicles are formed. Okay, so there can be intrinsic stimulus or extrinsic stimulus in case of apoptosis, and depending upon the what kind of a stimulus are involved, the apoptotic pathway leads in that way only. So I am not going into the details of these pathways because I hope you have understood it perfectly from the lecture. Still, if you have any questions, we can take them up, but first we should solve the assignment questions. So before going into the assignments, so conclusion is that apoptosis is a crucial process in multicellular organism. It's controlled and orderly form of cell death, and that plays a vital role in development, tissue homeostasis, and immune system. Because if cell death won't occur, then the immune cells which take up the antigen which are entering inside our body, then they won't die. And if they won't die, then that antigen along with our infected cells will become more dangerous for our system. So apoptosis is a must process which is required to maintain a harmony and homeostasis inside the body. Okay, so it is a very important process. This regulation of apoptosis is associated with various diseases. Okay, so it can lead to many serious illnesses. That's why it is important that a cell undergoes a programmed cell death. Okay, now we'll come to the questions of assignment. One by one we'll discuss the questions. Whoever hasn't understood any kind of any one of the questions can ask me, and the practice will go in the same way as we have been doing in the last lecture also. Everyone will type their answers for each questions one by one in the chat box only. No one will speak up the answers, so that whoever haven't got the answer can get time to tell their answers. Okay, so first of all we'll start with the question number 1. Question number 1 is, the primary role of insulin in our body is. Question answer for question number 1, everyone. The primary role of insulin in our body is. I'm waiting for your answers, everyone, in the chat box. Yes, the answer is B. To move glucose from bloodstream into the cells to produce energy. ठीक है? Okay, next question. Low blood glucose level causes what? Low blood glucose level causes what? Hanji. The answer to question number 2 is, yes, it is D. It will lead to the phosphorylation of pyruvate kinase. Okay, next question is, alpha ketoglutarate dehydrogenase is inhibited by succinyl coenzyme A and high levels of NADH, whereas following stimulates this step. What will stimulate this step? If succinyl coenzyme and high level of NADH is inhibiting alpha ketoglutarate dehydrogenase, what can activate it? We have discussed this also. So the answer to this question is calcium. Yes, calcium will help in the activation in stimulating this step. Okay, question number 4. In mammals, the primary site of beta oxidation for short, medium and long chain fatty acids is. बीटा ऑक्सीडेशन कहाँ होता है सेल्स में? Yes, the answer is mitochondria. Next question. Fatty acid activation takes place in the cytosol with the help of an enzyme. What enzyme? Fatty acid activation we are talking about here. Exactly, it is acyl coenzyme A synthetase. Next question. Following is the rate limiting step for fatty acid oxidation. कहाँ कहाँ rate limitation होती है? Carnitine shuttle. We have discussed this also. So it is the carnitine shuttle. Question number 7. G1 phase is under the control of what? ये डिस्कस किया हमने आज. It is cyclin D, because as I told you, every step has a checkpoint, and each checkpoint is involving what? Cyclin and cyclin dependent kinases. So it is cyclin D. Okay. Next question is, in following, nuclear membrane is dissolved and chromatin condenses into chromosomes. In which phase this happens? We have discussed it some time back only. Prophase. It is the prophase in which this chromatin condenses into chromosomes. Okay. Next question. Non-proliferative multicellular eukaryotic cells enter the dash phase. Non-proliferative multicellular eukaryotic cell enter the कौन से phase में जाती है non-proliferative cells? Yes, it is G0 phase. It is the G0 phase. Question number 10. The alignment of chromosome along with the metaphase plate is a crucial event to decide the entry of cells into the anaphase. The signal required for this control is created by what? Question number 10 का answer everyone. Yes, it is the mitotic spindle checkpoint. Next question. Cell cycle at different steps are slightly controlled by cell cycle checkpoints. Tightly controlled by cell cycle checkpoints. Each checkpoint is controlled by mutual interaction between what? We have discussed it in the two questions back. Question number 11th. Everyone. Yes, it is cyclin and cyclin dependent kinases. Okay. Next question is pycnosis is. What is pycnosis? Question number 12, everyone. What is pycnosis? Yes, it is a condensation of chromatin during apoptosis. Okay? Condensation of chromatin during apoptosis. Question number 13. One of the morphological features of necrosis is. Yes, it is cell swelling. Okay. Caspases selectively cleave proteins at site just C terminal to which residue? Which residue? Question number 14th. Question number 14th, everyone. Caspases selectively cleave proteins at site just C terminal to dash residue. Which residue? Aspartate. Yes, it is aspartate. Question number 15. The ratio between the following and pro-apoptotic proteins determines whether a cell lives or die. Ratio between what and pro-apoptotic proteins will determine whether the cell will live or die? Yes, anti-apoptotic proteins. Okay. So these were the questions of your assignment. Anyone who have faced difficulty in understanding these questions, or anyone who wants to discuss something else from last week's lecture, can ask me. We have discussed all the assignment questions. Anyone who wants to discuss anything else, who wants to ask something from the last week lecture, can ask me. That will be there on the NPTEL site only, Sakshi. Just check it once, and if you still won't get it, just contact to the NPTEL team. They'll help you. What happened with the connection? Hmm. IIT का Wi-Fi. So sorry everyone, my connection was lost. That's why I couldn't. I had to leave the meeting. So yes, anyone have any questions? If no one has any questions, then we can stop the session right here and we'll meet next week. So you guys can leave the meeting if you don't have any questions or you don't want to discuss anything from the last week's lecture. Thank you so much for joining.